This meeting is being recorded.
All right, folks. I see I'm still not. We're 4 out of the 8. Um, let's wait a couple more minutes still. I still have to upload something to your canvas real quick anyway. So we'll start in about 3 minutes, regardless. All right, there seems to be some issue with Moodle. I'm not sure what's going on there. Um, at any rate, I am the lecture that I'm going to go through today. I'm going to post the canvas right now so that you can have it to review. So I don't know what's going on with Moodle. I guess we'll figure that out over the weekend. All right. So that should do it. So remember, we're finishing up now the skeletal muscle um skeletal system today. And then we're going to move on to the muscle movement now. The actual means where the muscles can actually move. But before we do that, we have to, of course, finish with our major muscles that you need to know. Okay. Once again, these are not the only muscles that you should know, but the ones I'm going through here are the ones that are the most likely to show up on your tests, right? I think we had stopped yesterday at rectus femoris. We mentioned the rectus femoris, rectus, of course, because it's on the anterior, and femoris because it runs along the femur, right? So the anterior femur, and we mentioned this one here can flex the thigh, which of course means that you raise the thigh up towards your body, and it extends the lower leg outwards. So it's like you not jumping jacks, but high knees when you're running, and they lift the knees as high as possible when you're running in place. That is what I mean when I say they flex the thigh. And of course, extending the lower leg is like kicking, right? Now, this muscle here you may not have heard of before, the gracilis muscle. Now, you'll notice this muscle here is to be found on the medial aspect of the thigh, right? So medial, of course, closer to the middle, as you know, right? Now, based on the position of this muscle, I'm hoping it makes sense to you that this muscle can adduct the thigh. Considering where it is, remember, muscles can only pull, right? So consider it's where it is when it contracts, it should pull the muscle, pull the leg this way. So if you spread your legs and you want to stand up and you want to put them back where they were, this muscle, the gracilis, contracts and pulls your leg inwards, or as we say, medially, right? And it also flexes the thigh. So this one, along with the rectus femoris, help to lift your upper leg up, right? So of course that means that this muscle is important. Well, it assists in walking, right? So if you've ever sprained a muscle on your inner thigh while running or something like that, this might be the one that you hit, right? Now that said, that's not the only muscle on the inner thigh. But here's another one that you may have heard of before. This is a sartorius muscle. Now I want you to notice the angle of the muscle. The muscle is running from the iliac region and is then running across from the iliac region, as it says, the superior iliac spine. It then runs across to attach now to your leg, the medial surface of your leg, just below the knee joint. Okay? So considering where this is, and remember, your muscles can only contract. They can't push, they can only pull, right? Hopefully you can see that this one here is going to pull when this contracts, this pulls your leg that way, right? And what kind of movement is that? It says that it flexes the thigh, so it once again helps you to raise your upper leg and rotates the thigh. So by moving the leg this way, you can move out like that, right? That's what it means by rotating the leg laterally. You widen your legs or spread your legs if you prefer, however it is, right? So that's what the sartorius is for. So that's the movement you get from it, right? So if you go back to the gracilis, which is here, this one can only pull again, so it goes up that way, which is why it says it can adduct the leg. So it pulls the leg closer to the body, and this one can also help to flex the thigh, right? Then now we have the biceps femoris. We spoke about the biceps brachii yesterday. And the biceps brachii, of course, is an arm muscle. And it's called biceps because, of course, it has 2 points of origin. Just the same way the triceps has 3 points of origin, right? So, of course, the biceps femoris is also going to have 2 points of origin, but it is, of course, is associated with the femur, right? And this one extends the thigh and flexes the lower leg. So it extends the thigh, which of course is here, and flexes the lower leg. Because of course, if you look at the point of attachment, here's where you have your insertion. So it inserts below the kneecap and runs up, as you can see, the femur. And will then connect to your hip bone up here, which you can't see, right? So when this one contracts, it can pull the thigh upwards, which is the flexing, outwards, I should say, sorry, not upwards. So a can pull the thigh this way, or it can pull the leg that way. Okay? So extends the thigh and flexes the leg. All right. Then now we have this lovely one here called the gastrocnemius. Yeah, we can't see the 2 points of connection of the biceps brachii. It's connected to 2 points, and the idea here is that depending on where you pull, which one of the 2 points the muscle pulls from, you'll either move the whole hip, you'll either move the whole thigh, sorry, or you'll just pull the lower leg alone, right? So that's why it has 2 points of contact. So you can have differential pulling. Pull on one part, you move the thigh, pull another part, you move, of course, the lower leg, right? All right, now the gastrocnemius now. This one here is the one that you all call a calf muscle, right? You'll notice here it is the largest muscle you're going to find on the lower leg. And look where it's attached. It attaches, once again, over the knee joint. So it's going to be attaching to the femur somewhere in here. And it also runs down all the way to the calcaneal, which is the heel bone, right? Now, this muscle is one of the strongest muscles you're going to have in the lower leg, right? So people who do a lot of running, jogging, that kind of thing, sprinters, they normally have very pronounced gastrocnemius muscles, right? So when you look at the back of the leg, you can see it bulging out. That's the one that gives a sprinter power when they're sprinting. You don't normally see sprinters with little whingy gastrocnemius muscles, right? Also, because of the way it is attached to the heel, this one here is very, very, very susceptible to sharp blows right here. If you kick somebody hard enough right here, so it tears right through the gastrocnemius tendon. And when that happens, the muscle itself rolls up. I mean, it literally folds up and flies up over the knee joint. Like you know those blinds. You ever see? I'm sure you know blinds, right? When you pull up a pair of blinds, the muscle just rolls right up like that, right? And it is one of the most debilitating injuries you can get. People who play football are prone to this one because you go after the ball, somebody else go after the ball, you know, or they say they were going after the ball, you know, one kick and boom, the studs tear right through the gastrocnemius tendon. The muscle just flies up, and that's it. And this thing takes months to heal, right? And not only does it take months to heal, more often than not, it will not heal fully properly. So it'll end up with a gastrocnemius scar, and it will make the tendon shorter than it should be. And from that point on, when this person exercises, in no time they start to feel pain. And this is what you call a sports injury that will end your career. A lot of football players have had this, and that's it for them, right? So the gastrocnemius muscle here, or the calf, and you have the gastrocnemius tendon. Or I should mention that the tendon is also known as the Achilles tendon. Right? The Achilles tendon. Once that tear off, there's no more running for you. Right? Now, here is a muscle that helps you to wait, did I mention the guess what name? Should I mention the movement? So plantar flexion, right? So when it pulls on the heel, of course, your foot goes that way, right? It also mentions lower flexes foot and lower leg. So remember, flexing goes that way, right? So when you're running, this is the one that pulls your leg back, right? As you're making a forward step. So if without this, you just can't bend the leg at all, right? Now, the tibialis anterior now. As I said, this one dorsiflex and inverts the foot. So dorsiflex now means you're moving the foot up this way, right? So when you're taking a step, you normally lean your foot back a little bit. That's dorsiflexion, right? When you're going upstairs, anyway. Now, here is the location of the tibialis anterior. So it can pull the foot up. And that is so for the dorsiflexion, but it can also pull it up and laterally as well. So as in you try to look at the bottom of your foot, and that is the inversion part. So when you were looking at your foot bottom, you normally turn your foot medially towards yourself. So that is what it means by you're inverting your foot, right? All right. So as we said, those are not the only muscles by far, but we have to keep it reasonable. I'll be mentioning one or two other muscles as we go along. But I mean, how many muscles are we looking at? There's not even that many. We have here so from 82 to 103. That's not even 20 muscles, right? There will be other muscles, but please make sure you know at least these, because these are some of the biggest muscles in your body. They cause some of the largest movements. I guarantee that you'll be tested on some of these muscles, right? All right, now let's move on to the histology and physiology of your muscle, all right? So we're going to talk now about how muscles cause movement. Yes, we already stated that in order for you to move, your muscles have to shorten, right? Now, that said, how do muscles shorten? That's what we'll be looking at now. So remember, I posted this on Canvas. What name seems to be down? Moodle. I'm not 100% sure why. As a matter of fact, guys, can you give me 1 quick second? I just want to call my side real quick and see if I can find out what's going on with Moodle. So sorry, just give me one. Yeah, I can get through. All right. So this is life. I guess I'll have to wait until Monday to find out. Anyway, let's go back to this muscle business anyway. So as we see here, we have smooth muscles. The first part of this slide is just to remind you of the basic characteristics of the three types of muscle, right? So let's just run through it. It's good exercise to remember, right? So for example, it mentions here that smooth muscle fibers, which are the cells, remember, right, are shorter with a single nucleus, elongate with tapering ends, and the myofibrils are arranged randomly. What does that mean? So here, let me just draw one real quick. All right. This is a contracted smooth muscle fiber. There's my nucleus there. So it's short in comparison to the skeletal muscle. It tapers to both ends, so it's pointed at both ends, thickest in the middle. And myofibrils now are muscle proteins. And it says the muscle proteins in there run randomly. So you'll have the muscle protein sort of, as it says, randomly. And this is important because it actually impacts the strength of the contraction in any one given direction. But it means that overall, in multiple directions, the muscle can still contract, right? So it might not be super strong in contracting in one direction, but it can contract in multiple directions. It has no striations. So for example, skeletal muscle and cardiac muscle would be striated. And it lacks transverse tubules. This here, I'm going to explain what this is to you shortly. And the sarcoplasmic reticulum is not well developed. Now, you're going to have to get used to the term sarco. Sarco is muscle, specifically the muscle cell, right? A lot of structures in muscle cells, of course, are going to be in everyday normal cells. But to differentiate them from everyday normal cells, we just call them sarco. So, for example, the cell membrane of a muscle cell is not called cell membrane. It's called a sarcolemma, right? The cytoplasm is not called cytoplasm. It is called sarcoplasm. I'm assuming you all see where I'm going with this, right? The sarcoplasmic reticulum that sarcoplasmic reticulum it mentions here, in all other cells, we'd call it endoplasmic reticulum. Right? So when it comes to muscle cells, you basically just put in sarco in the words that are normal things that the muscle cell has. I'm surprised the nucleus isn't called a sarculus or something. But so sarcolemma is the cell membrane. Sarcoplasm is the cell cytoplasm, and sarcoplasmic reticulum is the endoplasmic reticulum of the cell. Remember, this is for the muscle cell only, right? Now, this here now indicates the function of the smooth muscle cell. It's for peristalsis, which is rhythmic contractions that push substances through body tubes. We all know peristalsis from C-sec, right? Now, the smooth muscles themselves are going, certain neurotransmitters are going to be used to activate those muscles. We have acetylcholine, norepinephrine. Okay. So it says these are the neurotransmitters for smooth muscle contraction. Now, cardiac muscle, which is heart muscle, spoke about this one already. As it says, muscle fibers are joined together by intercalated discs. Fibers are branched. So once again, let's do a quick little sketch of that. Another cell down there, yeah? All right. So this is a smooth muscle cell. It's relatively short and it's branched, as you can see here, right? Now, and it has intercalated discs. So this is your intercalated disc. Now they tend to have large nuclei, okay? But here's the thing. It can have one or two of them. And it is striated. So when you look in there, it's going to look striped. And it's a straight, why not? This is just a rough sketch, right? Very rough. Very, very, very rough. All right, you get the idea. Now, we can compare that, of course, to that. So the smooth muscle looks very different from the cardiac muscle cell, okay? Remember, it's longer than a smooth muscle, but it's by no means a long cell, right? It's branched. It's the only cell that we have so far that is actually bifurcated, split in two. You find it only in the heart. No surprise, there's a cardiac muscle cell. And you have the intercalated discs running between the cells, right? And these intercalated discs are used for signal transmission. Now, it mentions here that the cell is self-excitatory. So basically, it can send its own signal to cause itself to contract, right? And we spoke about refractory periods already in ENP1 and about basically it's the time it takes the cell to recover from a contraction. So when the heart muscle contracts, it takes longer before it can contract again than smooth muscle or skeletal muscle. This, of course, is to give time for the filling of the ventricles and the atria, etc., right? Now, the last one, which is, or I should mention, acetylcholine is the neurotransmitter that we use in the cardiac muscle as well. Now, if you notice here, acetylcholine slows the heart rate, whereas norepinephrine speeds up the heart rate, okay? So these two are also used, remember, in your smooth muscle. So the same idea, acetylcholine speeds up the heart rate, norepinephrine slows it down, right? So it's a bit of, so it's opposite to what we have here in the heart, right? Now, the last one, of course, is the skeletal muscle. As we know, it's most of the muscles in the body. Most of the weight of the body, well, 40-50% of the total body weight can be muscle. And this one now, as we know, this one is the longest muscle cell. This one is very, very long, remember. Sometimes centimeters long. This one is striated. Very similar to what's going on in the cardiac muscle. We're going to talk about the striations in detail. And of course, the nuclei. Remember that the cardiac muscle could have one or two nuclei. These lung cells here are multinucleate. So they could have dozens of nuclei in the cell. And they're found near the periphery of the cell as well. All right. So that's your skeletal muscle. As it says here, their main function is force production for locomotion and breathing. So smooth muscle is for things like peristalsis, helping to move substances along through tubes. Cardiac muscles help to pump blood around the body, as we know, and skeletal muscles for general movement, locomotion, breathing of the body, right? So there we have our comparison of the three types of muscle. Now, what about the neurotransmitter used in skeletal muscle? That is going to be acetylcholine, right? So the more acetylcholine you have, the stronger the skeletal muscle contracts. Unlike what happening in your heart, whereas you can see your acetylcholine actually slows the heart down, right? All right. It also mentions here that with skeletal muscles, once you release acetylcholine and cause that muscle contraction, you have to get rid of the acetylcholine before the muscle can relax, which is why you need an enzyme here called acetylcholine esterase. And it, of course, breaks down acetylcholine and removes it from the skeletal muscle, allowing that muscle to relax. So when you make a fist or something, you're contracting your muscles, right? And they stay contracted. Once you decide you want to relax your hand, then you have your body releases acetylcholine into the synapses, which we'll talk about later, and break down acetylcholine so fast, it takes you a fraction of a second to relax that muscle, right? So fast it goes. But you still have to break down, but it's not instantaneous because you have to get rid of the acetylcholine before that muscle can relax, okay? So you can't instantaneously relax a muscle. Now, we're going to talk now about the covering of the skeletal muscle, all right? We mentioned fascicles earlier when we were doing the muscle classifications and how fascicles are groups of muscle cells, right? And they're surrounded by a membrane. We didn't really get into what the membrane was. So at this point now, we're going to talk about the membranes associated with the muscles. So for example here, or I should say, first one we're looking at here is the epimysium. And as usual, whenever you see epi, you know it's referring to outside of something, right? So the epimysium covers the entire muscle. So it's a connective tissue layer that wraps around the whole muscle, epimysium, okay? And then now your individual fascicles have a perimysium around them. So the epimysium surrounds the perimysium. And then now each individual muscle cell has its own little membrane around it called a endomysium. So epimysium around the whole muscle, perimysium around the fascicles, and endomysium around the individual muscle cells, right?
Can you repeat that, sir?
All right. So epimysium is the connective tissue layer you'll find around the whole muscle, the whole thing. Then perimysium, you'll find that around the fascicles. Remember the fascicles are we spoke about when we're doing muscle shapes. And endomysium is going to be around individual fascicles, right? So think of it this way. When we're doing, for example, the biceps, which of course is a parallel muscle, we could see muscle groups in there like this, right? And what we'd end up having is the epimysium epimysium would be the membrane right here, surrounding the whole muscle. Okay. So it essentially wraps the muscle like cellophane. And that would be the epimysium. Now, if you cut this muscle, right, you did a cross section of this muscle, well, you'd have something sort of like this. And so you have little sections here. And inside the individual sections, you would see the ends of the muscle fibers that are cut. So that would be like the muscle fibers that you're looking straight at. And now this membrane around it here now, that would be the perimysium. You know, let's do it the easy way. All right, let's do this again. That's the cross section to the muscle. These are our muscle bundles here. And then inside that now, you will have your fascicle, your individual muscle cells, right? All bundled up on the inside. And what we're saying is that that membrane that is around the individual bundles of muscle, that now is the perimysium, okay? So each one of these is wrapped around with a perimysium. But when you look at the individual muscle cells themselves, so if I take out one of these little blue ones and put it out here, so let's say that's the muscle cell. It in itself has a membrane around it. And that membrane now is going to be your endomysium. So it is surrounded by endomysium. That will be the endomysium, right? So once again, epimysium wraps the whole muscle, perimysium wraps the fascicles, and endomysium wraps around the individual muscle cells, right? What's going on? We're going from four to three. What's going on? Anyway, now just to remind everybody about origin and insertion. Origin immovable part of the bone. That's what when the muscle contracts, that bone does not move. Insertion, on the other hand, movable part. When that contracts, when the muscle contracts, that bone moves. Right? Now, here are some terms that I don't think we've touched yet, which are prime mover, synergist, and antagonist. Okay? Your prime mover is the main muscle related to the movement. So your prime mover does most of the work, basically, right? So, for example, here, when you're trying to do a curl, right? You're flexing your elbow joint or your arm. The prime mover here is going to be the biceps brachii. This is the muscle that does most of the work. That's what makes it prime, right? But it is not the only muscle that is working. So, for example, it mentions here that the prime mover might do most of the work, but it can also have synergists with it. And a synergist basically is a muscle that assists the prime mover. It helps it to do its job. So the deltoid muscle, which remember the deltoid sits up here, right? So the deltoid is up here. So when the biceps brachii contracts and pulls on your forearm to bring your forearm forward, remember we're going in this direction, so we're flexing. The deltoid isn't pulling on the forearm. What the deltoid does is it stabilizes the arm so the arm doesn't wobble all over the place when you're trying to do a curl. So is it the one causing the flexing? No, but it makes the flexing easier by stabilizing the whole arm, right? Then now you can also have what we call antagonists. And I'm sure everybody has been antagonized at least once in their life. So you know that an antagonist is somebody that is going against you, right? And for antagonism, as it says here, it resists the prime mover's action and causes an opposite movement. So the prime mover causes a movement in a particular direction. The synergist helps the movement to occur, and the antagonist does the exact opposite of that movement. So, for example, the biceps brachii will, of course, as you know, flex. So the other muscle now would be the triceps brachii. I'm going to draw all three parts of it. But as you know, this one is also connected. So the triceps brachii, when it contracts, it will cause the arm to extend. Right? As you notice here, it's on the opposite side of the biceps brachii because it causes opposite motion, right? So it is an antagonist. Whatever the biceps is doing, the triceps is undoing, right? All right. So here we have, of course, our epimysium, perimysium, etc., right? Which you spoke about earlier. Okay. So now that we've gone through, and we don't remember the basics. This is once again just information to remind you all, right? But I just went through it. So let us talk now about in more detail now about the structure of your skeletal muscle. So we're going to put a little bit more detail into this one. Because of course, right, once again, when we say muscular system or musculoskeletal system, the muscle we're referring to is going to be your skeletal muscle, right? So we mentioned sarcolemma already, is the cell membrane, right? And I think I also mentioned to you that myofibrils are muscle proteins. So myofibrils are muscle proteins. Generally speaking, They are, as it says, thread-like strands within the muscle fibers. Remember, the muscle fiber is the muscle cell, right? So it says fiber, but it's really the cell they're talking about. So you have thread-like strands inside the muscle cells. For the most part, it is these thread-like strands here that are going to cause contraction. So these are the ones that will actually cause movement, cause things to happen. And they are also the ones that give strength to that contraction. Simply put, the more myofibrils you have in your muscle cell, the stronger that muscle cell is. Okay? All right. Now, it mentions here that the myofibrils have two main parts. You have one part is called an actin, also known as a thin filament, and it is made up of two proteins. One is called troponin, another one is called tropomyosin. Okay? So your myofibrils, two major proteins or two major parts. You have one that is called the actin, also known as a thin filament, and it is made up of two main proteins. One is called troponin, the other one is called tropomyosin. And the other part now is called myosin. And it is also known as the thick filament. Right? So fundamentally, your myofibrils are comprised of a thick filament and a thin filament. And remember, the myofibrils are going to cause movement, right? So obviously, thick filaments, thin filaments must be involved some way, somehow, in causing movement to occur, right? So this here now is actually a section through your muscle cell. As you can see, it looks as if it was peeled back to reveal all the lovely innards, okay? Let's just point out sarcolemma first. Remember, that's a cell membrane. Then we can see the nuclei pressed up against the inside of the sarcolemma here. So remember, these nuclei, even though they're on the surface, they're actually pressed into the sarcolemma from underneath. That's why it looks like that, right? Within the muscle cell itself, here we now have our myofibril. So this whole thing here says a myofibril. And I want you to notice that we have to find now thick filament and thin filament in that myofibril, right? Because remember, those are the major sections, right? So it shows here a section of this, a section here, right? Of your myofibril. And this section is called a sarcomere. Sarcomere is very important. The sarcomere is a functional section of your myofibril. Okay? It is the smallest part that can do its job. And the job, of course, is contraction. So this sarcomere here is the smallest part of the myofibril that can actually contract, that can cause movement, right? So what we're going to do is we're going to examine the sarcomere in a little bit more detail, and we're going to look now for filaments, okay? So when you look down here now, all right, you know what, let me put this in another slide. Just to make it a little bit bigger, let's put it on another slide. So here we have it a little bit bigger. So remember now, our myofibrils, those are the thread-like structures within the muscle cell. They're the ones that will contract, okay? They themselves are made up of proteins, two major proteins. We have actin and myosin, also known as thin filament and thick filament. But the thin filament and thick filament are arranged in a certain way to form sections, right? So you can think of a long rope with sections cut into it, okay? Each section is called a sarcomere. And each sarcomere has everything required for contraction to occur, which is why we call it the basic unit of contraction, okay? Now, the ends of your sarcomere are going to be known as Z discs, all right? So the Z disc is where one sarcomere ends and another sarcomere begins, right? So the idea is that on either end of the sarcomere here, you're going to have a Z disc, which connects this sarcomere to the one right beside it, okay? Remember, we're arranging these things in a line, basically on the thread, right? Now, within the sarcomere is where we're going to find now our elusive thin filaments and thick filaments, right? So if you look now, so remember, all we've done is we've cut out this sarcomere here, and we've enlarged it so it's kind of more easier to see. Within here now, here's our Z disc, but we have now found our filaments. So if you can see here from the one on top, you can see horizontal lines running through. All you could see are lines. You really couldn't see what was going on with the lines. All we know is that some lines were blue and some lines were, I don't know, orange, whatever color that is, right? So when we blow it up now, we can see here now our orange lines here, they look orange to me, are the thick filaments. So these orange ones are thick filaments. And then the blue ones here are the thin filaments, okay? Now look at the way we have these things arranged. You know what, I'm assuming everybody knows what a soul comb is, right? So think of it like a soul comb. So we have our Z discs, like the comb handle, and then we have our thin filaments coming off of it, like the teeth of the comb, right? Right? So those are the thin filaments. And in between now, the thin filaments, in between our comb, you have the thick filaments sitting. And of course, they're thick filaments because they are broader. So they're sitting on the inside like that. Right? And they run, of course, from one side of the sarcomere to the next. But I'm just showing you here the basic idea, right? So there they are. And you'll notice, of course, that there is a little structure running down that sort of holds them together. It's not actually a structure. We call that the M line. Anyway, so hopefully that this makes sense to you now, right? So we have the thin filaments attached to the Z discs, just like a comb, as we said, the teeth of the comb. M line, as in mango, right? So the M line you can see here, your thick filaments are coming off of the M line, running in between the thin filaments, okay? And the thin filaments are attached to the Z disc coming off like the teeth of a comb. So that means, of course, that your thick filaments and your thin filaments are essentially overlapping, right? Now, how does this cause movement? This doesn't look like it would cause any movement. This looks like two sole comb and some bobby pin. So how does this cause movement? That's the next thing we have to talk about now, right? But just remember, this is the basic setup we have here. We have our Z discs on either side of the sarcomere, thin filaments coming off of them, thick filaments running in between them, right? That is the basics, okay? I'll talk about the A band and everything like that and the I band in a little while, right? Just take this in for now. So I ask you to draw a section of sarcomere. This is what I'm essentially expecting to get, something like this. It doesn't have to be fancy. It can be something as simple as what I have over here, right? All right. Now, in writing now, says the further divisions of the myofibril are the Z line or Z disc, same thing, A band, I band. So I'm going to have to explain what the bands are in a second. Within the sarcoplasm, which remember is the same as cytoplasm, you have the sarcoplasmic reticulum, which is the same as endoplasmic reticulum, right? Remember, sarcoplasm is the same as cytoplasm, and sarcoplasmic reticulum is the same as endoplasmic reticulum. Now, as you know, as endoplasmic reticulum in your regular cells, well, depending on the type of endoplasmic reticulum you have, is pretty much for production of something, right? So if it's rough endoplasmic reticulum, you know you're making something protein-based. If it's smooth endoplasmic reticulum, you're making something carbohydrate or lipid-based, right? But in your muscle cells, the sarcoplasmic reticulum doesn't do any of that. The sarcoplasmic reticulum's job is actually to store calcium, right? So it's not making anything, it's keeping calcium sequestered. You all know the word sequester, right? You're keeping it away from something else. So it's not a matter of how you're hanging on to it. You're sequestering it. You don't want it in the cytoplasm, so you put it in the sarcoplasmic reticulum. And you'll understand why we do it this way in a little bit, right? Now, it also mentions that you have these things called T-tubules, right? And terminals, which are transverse tubules, and terminal cisterna, right? If we go back to the original image here, we can see we have these yellow T-tubules, and then the cisterna are the bulging parts of the sarcoplasmic reticulum, which is the green stuff, right? So the T-tubule runs along the cisterna, right? Now, the T-tubules are going to be very important. They are going to sit on the outside. Well, they will come from the outside on the sarcolemma. So the tube here ends on the surface and then runs down from the surface along the cisterna of your sarcoplasmic reticulum. Okay? All right. So what I'm going to do is I'm going to try and explain how a signal is sent that is going to start the process of muscle contraction, right? So I'm going to do a very, very simple diagram first, as usual, and then we look at what we have on the slide so we can do a comparative, right? That's how we usually do, right? Remember, It's going to be a simple diagram. But as usual, if I give you a diagram, even if it's a simple looking one, it's because it will be adequate to explaining something. And if you do a diagram like that, if you're asked to do so, that diagram is perfectly fine, right? As simple as it might be, doesn't mean it's not useful, right? Okay. All right, so let's start with this. This here is going to represent now my sarcolemma. Remember, my sarcolemma is the same as my cell membrane, right? Okay. Now, coming out of the sarcolemma, we're going to have these T tubules. And they'll run down from the sarcolemma into the cell itself, like that. I'm just trying to show it running through the cell membrane down into the cell. So remember that's my T tubule. Now my T tubules are running over my sarcoplasmic reticulum. Specifically, they're in a thicker part of it called the terminal cisterna. And then running between them now, you're going to have the other aspects or the other parts of the sarcoplasmic reticulum. Remember, it's a simple diagram I'm doing, right? Nothing fancy. I'm not showing connecting tubules here, right? So remember now, this is my terminal cisterna. And the whole thing, so the terminal cisterna, remember, just the part right here, so where the T tubule is running. All right. And the whole thing, of course, is my sarcoplasmic reticulum. Okay. Now, below that, we're going to have our we're going to draw our sarcomere. Obviously, I'm oversizing the thing, okay? It's way smaller than this in reality, in comparison, but we're doing this to make it easier to see what's going on, right? And of course, we need, so those will be our thin filaments. And these will be our thick. All right. So remember this here, sarcomere. So that's our section of myofibril, right? Okay. So we have the basic setup here. So how is this going to work? In order for the muscle to contract, it has to receive a signal, an electrical signal or a depolarization, however you want to put it, okay? Now that signal has to hit the muscle cell, the sarcolemma, the cell membrane, right? So that one is going to come across a synapse. Everybody remember synapse, right? So the synapse of muscle cells is called a neuromuscular junction. Neuromuscular junction. Because why make things simple, right? So across the neuromuscular junction, which is fundamentally the same as a synapse, you release a neurotransmitter. The neurotransmitter moves over and attaches itself to the sarcolemma, okay? That sends a signal down the sarcolemma membrane. That signal runs along until it's a T tubule, and that signal gets carried inside the cell, okay? And that signal inside the cell, I'm gonna keep going to the next one, of course, is going to cause now the release of calcium. Remember that the sarcoplasmic reticulum store calcium, right? So once the signal now hits the sarcoplasmic reticulum, you start to dump calcium. Okay? And that calcium that the sarcoplasmic reticulum dumps is going to now cause the muscle to contract. Okay? And it's going to contract pretty much all the sarcomere are going to contract at the same time, because remember, the myofibrils are floating in the cytoplasm. You've now dumped calcium in the cytoplasm, which means the calcium is going to go to all parts of the myofibril at the same time. So all the myofibril in the cell are going to contract at the same time. All right. Any questions so far, or so far so good? Should I run back through this part again one more time, or can I move on?
Sir, can I run to one more time?
Okay, one more time. So remember now, what I'm showing here is simply how the muscle contraction is initiated, okay? I haven't actually showed you how the sarcomere contracts yet. I'm not showing you what causes it. So I'm saying a muscle cell, remember, this is a skeletal muscle cell. A muscle cell membrane, which is the sarcolemma, is going to receive a neurotransmitter, right? And the neurotransmitter is going to bind to the sarcolemma. So remember, this is the neurotransmitter here. Goes down to the cell membrane, which is the sarcolemma, remember, binds to the sarcolemma, and that triggers a signal or a voltage, or all right, however you want to put it, the same idea. That signal or voltage runs down the sarcolemma until it hits the T tubules. And the T tubules run from the surface of the cell membrane, the sarcolemma, all the way down into your sarcoplasmic reticulum, right? And they're found especially in parts of the sarcoplasmic reticulum that store calcium, called terminal cisterna. Okay? It's basically a big bag of calcium. When the signal runs down the T tubule, it causes the terminal cisterna of the sarcoplasmic reticulum, remember, it's a part of it, to release calcium into the cytoplasm of the cell or into the sarcoplasm, since it's a muscle cell, right? And then that now, calcium is going to interact now with the sarcomere and cause the muscle to contract, okay? So the next step of this now is to explain how calcium causes muscle contraction. All right. So far, so good. Can we move on?
Yes, sir.
Okay, cool. All right. Now, the calcium, okay, I think I need another thing here. All right, I'm just gonna do the next part on this here. Might make it a little easier for me. All right. So, what does the calcium actually do? Remember, we spoke about the thin filaments and thick filaments, right? I just drew a sarcomere a little while ago, and of course we're gonna redraw it again now. All right. Anyway, let's do it again real quick. Remember, Z disc or Z line, whatever, doesn't matter. Thin filament. And in running in between them now, you'll have the thick filament, right? That, of course, is my thick filament. Now, when we look at the thin filament in more detail, you're going to see that the thin filament has two parts. We mentioned them already, which is, of course, your troponin and your tropomyosin, okay? Yeah. I'm just making sure something is here real quick. Okay. Anyway, let's go back. So we're going to look now at the thin filament in some detail. I'm going to, of course, simplify as usual, right? So your actin, pretty much think of your actin molecules as little spheres like this, okay? So they're little proteins that are like little balls, right? There shouldn't be any spaces between them where you get the idea. Now, ordinarily, these little balls of actin are separated from everything by tropomyosin. And tropomyosin acts as little strands that block off the actin from anything touching it, right? So they wrap around the actin to keep it separate from everybody. And by everybody, of course, I mean the thick filament, which we'll talk about in a second, okay? But your tropomyosin, sorry, has binding sites on it. called a troponin. And of course, these binding sites are for calcium, right? So remember, this is the basic setup of your thin filament. You have the actin here. Wrapped around the actin is your tropomyosin. And on the tropomyosin, you have a calcium binding site called a troponin. Okay? So all of this, remember, is thin filament. Okay? Hopefully so far so good. Now on the thick filament now, let me show you the setup for that one. The thick filament is simpler. Oh, it disappears on me. Yeah. Thick filament is simpler. You're not going to have all of that confusing stuff on the thick filament. Instead, what you're going to have is what we call a head. It kind of looks like a golf club. I'm assuming you all know what golf clubs look like, right? You know what? I want to change the color of this. So it has a head, kind of like a golf club. It has a hinge in it. And then it has the rest of the body of the filament. All right? So here's my basic setup. Okay? So this is my myosin head. Okay? That is my filament. And that part here is my hinge. And that makes up the thick filament. Obviously not to scale, right? All right. Here's the basic idea. The myosin here wants to bind to the actin. The head of the myosin here wants to grab onto the actin, okay? But of course, the tropomyosin is in the way. So what we have to do in order for the myosin to reach up and grab onto the actin is to get the tropomyosin out of the way. And that is where calcium comes in. Calcium is going to move in, bind to the troponin. Remember, that's a calcium binding site. And when it does that, it pulls the tropomyosin out of the way. That now exposes the myosin and the actin. And when that happens, the myosin actually lifts up and grabs on to your actin. All right, let me get one more slide. Remember, I do these diagrams so that you can do them yourself if you need to, right? Because the slide itself, there are very nice diagrams on it, right? But of course, you can't draw those. So once the calcium comes in, remember that we're going to move the tropomyosin out of the way. And now we're left with the actin exposed. Okay? So we'll say that's calcium. So now we've moved the tropomyosin out of the way. Here now the actin is now available. When that happens, that is now when you're going to have, should be more consistent with the colors, but whatever. The myosin now coming in and doing their thing. So remember now, the myosin head is going to reach up and it's going to bind to the actin. Remember, we have a hinge here. So it reaches up and grabs the actin, okay? And once it does that, it is going to pull in that direction. So the head is going to bend more and pull the actin, right? And as it pulls on the actin, remember, we're talking about millions of these molecules all doing the same thing at the same time. So you'll have millions of myosins bound to millions of actin, all of which are going to be occurring within now the sarcomere. And so all of these myosin here will be bound to the actin up here at all of these levels. So millions of them are bound. And all of them are going to pull at the same time. So all of these millions of actins bound to the millions of myosin, the myosin heads will bend and pull at the exact same time. And as they bend and pull, the Z discs get pulled towards the middle of the sarcomere by the myosin heads bending and pulling on them. Okay? And there's an imaginary line running down the middle of the sarcomere, which we call the M line. It's not really there, but you imagine that it's there. So the idea here is that you're pulling now the Z discs closer together, right? And as you're doing that, you're pulling the thin filaments closer to the M line. Does that make sense so far?
Yes, sir.
Okay. So remember now, you have a Z disc on either side. So it's like you have two combs. You have the thin filaments coming off on both sides. The myosin reach up and grab onto the thin filaments and just yank them towards the middle. So of course it means that you're pulling the Z disc that way. So that of course means that the sarcomere is going to get shorter because you're pulling in those Z discs. Okay? Now the H zone here is the area where you only have thick filament. All right, so this area here, only thick filament is there. There's no thin filament overlapping. So like over here, so you have thin filament, then thick, then thin, then thick, etc. Right? Only near the middle are you going to have only the thick filament and no thin. But once we get the contraction of the myosin going on, you're going to yank now or pull the thin filaments closer to the M line, which now means your H zone is going to shrink because there will be now more overlap between the thin filament and the thick filament and less space where you have the thick filament alone. Because remember, you're moving the Z discs and therefore the thin filaments closer to the middle, so there will be more overlap, okay? Now, in terms of the A band here is the area of overlap between the thick filament and the thin filament, right? All right, let me just make this a little bigger too. Crop copy. All right, just to make things a little more obvious, right? So remember now, we have our H zone, which is just the area where you have thick filament alone, right? We have our A band, which is where you have the overlapping of the thin and the thick filament, okay? So remember now, the thick filament reaches up, grabs onto the thin filament, and pulls the thin filament towards the middle. And the middle, of course, is where you have our M line here. So, of course, as you're pulling, the Z discs here are going to move closer together, right? And as that happens, of course, the H zone is going to narrow because you're moving the thin filament closer to the middle. So you have less and less space left without thin filament overlapping it, right? And because of that, of course, the sarcomere itself has to get shorter because the Z discs are moving closer together. Okay? Now, the other band is the I band. The I band is the space where you have only thin filament. Okay? So on either side of the Z disc, you have this area here with no thick filaments at all. That is the I band. In the middle, remember, we have thick filaments alone. That is the H zone. We have them overlapping one another. That's the A band. Okay? The line that runs down the middle of the sarcomere is the M line. All right. So if you see this guy, you should be able to label it. Okay? Anyway, remember now, by pulling now the Z discs via tugging on the thin filaments, you're shortening the sarcomere. Now, remember that calcium is being released into the sarcoplasm. So all of the myofibrils are getting calcium at the same time. Remember what the calcium does, right? The calcium pulls the troponin, the calcium binds to the troponin and pulls the tropomyosin out of the way, leaving the actin free. Once the actin is free, the myosin reaches up and grabs on now. So let's use what's in the PowerPoint now, right? So here we go. Tropomyosin wrapped around the actin, troponin head. Calcium comes along, binds to the troponin, pulls the tropomyosin out of the way. Now, the myosin down here can now get to the actin. So the myosin reaches up and is going to want to bind to that actin, okay? Let's ignore the ATP for now. So the myosin head then binds to the actin, and once it binds, it pulls. So you can see the direction of pull is that way. That is towards the M line. And remember, millions of molecules are doing this, if not billions at the same time. So all of them grabbing on to the actin, and they're all pulling at the same time, which results, of course, in the thin filaments pulling the Z, being pulled along and pulling the Z discs along as well, right? As long as calcium is present, the actin is going to be exposed, so the myosin will always reach up, grab and pull, reach up, grab and pull. It's like you're pulling a rope, right? So one hand over the next, constantly pulling, which means, of course, as long as there's calcium, the sarcomere is going to want to shorten because the myosin is going to want to pull on it, right? And remember, that results in the shortening of the sarcomere. And if you shorten the sarcomeres, you're going to shorten the myofibrils. And there's thousands of them in a muscle cell. And they're attached to the cell membrane of the muscle cell, the myofibrils. So when you shorten the myofibrils, you also shorten the muscle cell, okay? And when you shorten the muscle cells, you obviously shorten the muscle as well. And as you shorten the muscle, what does the muscle do? It pulls inwards. So whatever is attached to the muscle gets pulled as well. Hence, you get movement. So it is a shortening of the muscle cells that will lead to the shortening of the muscle. And the muscles only shorten because the myofibrils are shortening. And the only reason the myofibrils shorten is because the sarcomeres are shortening. All right. Any questions there so far? Okay. All is well, good. So remember, if I ask you to draw a diagram, you can do something as simple as that and explain it, right? You don't have to do anything overly complicated. But so keep that in mind. But at the same time, I might simply ask you to draw a section through sarcomere, indicating the important regions, in which case, of course, you're drawing something like this, right? And then you will put in the regions. Or, of course, could do something as simple as that. And then you could put in the regions here as well, right? Because, for example, here, we have A band, which is overlapping of the actin and the myosin. We have the M line here. We have our H zone here, which is just the myosin alone. As of course, we have our Z discs on either side. And we have, of course, our I band. Okay? So you can do something as simple as that. If you're asked to draw a sarcomere. And that's the work of like a minute or two, right? So keep in mind, your in-course test could have structured components to it. You could have diagrams to label. You could or interpret. You could be asked to, with the aid of a diagram, do whatever, right? So you need to be able. So the things you see me draw, I'm not drawing them because I like to doodle. I'm drawing them to show you how simple a diagram can be and still be useful. Okay? So please keep that in mind, right? All right. No more questions, so let's continue. We have another, well, 3 or 4 minutes. That's all right. That's good enough. Okay. So remember now, it is the contraction of the sarcomere that leads to the muscle contraction overall. And we explain why. And remember, you need to be able to label the sarcomere. Also remember, as long as there is calcium present, then the troponin will bind to the calcium and stay away from, and pull the tropomyosin out of the way, exposing the actin. As long as the actin is exposed, the myosin is going to want to attach to it and pull on it, right? So therefore, if you want to relax a muscle, you must be able to get rid of the calcium that is in the cytoplasm so that the muscle can relax, okay? So if there's plenty of calcium in the muscle cell, it's going to be contracted if it's in the cytoplasm. We'll talk about it a little bit more. Instead, now I want to come back to this diagram here and talk about the ATP aspect of it, okay? So let's talk about the ATP. We already know about the calcium release, right? So this diagram here is showing signal comes down a nerve, signal goes to the sarcolemma, which of course is, as you know, the cell membrane of the muscle cell. So this is showing the neuromuscular junction. Remember, neuromuscular junction, same as a synapse basically. The signal runs down the neuromuscular runs from the neuromuscular junction down the sarcolemma, down the T tubule, which I showed you earlier in another little diagram. And that signal then moves into the sarcoplasmic reticulum. You release calcium, right? Remember, the release of the calcium is where you get the calcium from to do this. To bind to the troponin and pull the tropomyosin out of the way. Now, at the same time, I want you to look down here. Notice that your myosin, which is lying down nice and relaxed, contains a molecule of ADP. Contains a molecule of ADP and phosphate. Where you get ADP and phosphate from? Of course, you get it from ATP. Right? So the ATP is broken down by your myosin and it binds to the myosin. Okay? When it binds to the myosin, the myosin comes into the ready position here. So can you see this myosin at the side here is just lying down, minding its own business. It's not doing anything much. Versus this one. So this one is kind of like that. Right? Just lying there, not doing very much. When you bind the ATP, right, to it, you get this. And we say that the myosin here is cocked. You know, like a gun, you pull back the hammer on the gun, so the gun is cocked, right? That now means the myosin here now is ready to bind, okay? So once you have cocked the myosin with the ATP, remember it binds to it, the ADP plus phosphate bind to the myosin head. You then expose now the actin. Once you do that, the cocked myosin reaches up, grabs on to the actin. So remember, you need the ADP for this to work. Once that happens, once you grab on, the ADP is released from the myosin. So the ADP and phosphate get released. And once they let go, then now, remember, we have a hinge right here. The release of the ADP and the phosphate from the myosin causes the hinge to bend inwards. So that is what causes you to pull on the thin filament, okay? So you release the ADP, and of course, it's like pulling the trigger. The ADP fly out like the bullet, and the myosin pulls on the actin, right? And of course, this means you're moving the Z discs closer to the M line, causing the sarcomere to shorten. Now, once you pull, you can see the myosin releases the actin, right? And it goes back to its original position, where it binds again with another ATP, to get the ADP and phosphate in there. And as long as the actin is exposed, it's going to reach up again and grab again, right? So it grabs and pulls, then lets go, then grabs and pulls again. So it's like as I said, it's like you're pulling on a rope, hand over hand, one hand in the next, then the next, pulling something towards you. So the myosin reaches up, grabs, pulls, then lets go, reaches up, grabs again, then pulls. So the idea is that it's grabbing and pulling faster than the sarcomere can run back to its original position, right? So the sarcomere, if you let it go, it just slides back out to its original position. But as long as you have the myosin working, it keeps getting pulled towards the M line. So that means that now that the sarcomere stays shortened and the muscle stays contracted, okay? Now, if you run out of ATP, you can no longer cock the myosin head, which means it can no longer bind to the actin. Without ATP to power this process, the sarcomere just runs back to its original position and your muscle goes from a contracted state to a relaxed state, right? This is why you need to have ATP during muscle contractions. It is what sustains the contraction. So if you're working out, as we said, I mentioned muscle failure. When do muscles fail? You run out of the ATP to power this process. Once you run out of ATP, the muscle just runs back to its normal position. It has failed, and your muscle essentially stops functioning for contraction's sake, right? The other way of causing problems for this is to remove the calcium from the muscle cell. If you pull the calcium out, all the ATP in the world is not going to make the muscle contract, which is one of the reasons why we store calcium in our bodies, because lack of calcium means your muscles don't work. And if your muscles don't work, you are dead, right? Simple as that. You're dead. So that is why we store calcium in our bones, to prevent the whole you are dead thing from occurring, right? Okay. So hopefully this makes sense to you all. I will, of course, run back through it again next week, Tuesday, or whenever it is. But of course, what I just described is here in word form. So please read through it for me, please. And if there's anything that you don't understand, try bring it up to me next time when I run through it, okay? All right, it's 6:50, so I'll stop here. But we should finish muscles next week. And what's the next topic? I need to go check. But I think what I'm trying for, let's try for the 3 topics. Then we do our in-course test. So we're doing muscles, skeletal system. Oh, no, wait, the next one is nervous system. I might swap out the nervous system and leave it a little bit, because the nervous system is a pretty big topic. I wouldn't necessarily want it all big topics on the first in-course test. So next week we'll figure out what we're doing. I will post the next PowerPoint as well, so we have it used for next week. Otherwise, folks, we can stop here.
Sir,
Yes.
you said you're going to post some like worksheets, like for practice?
Yeah, I'm gonna have to post them on Canvas. I was hoping to do it on Moodle, but I don't know what's going on with Moodle. So I'll post them on Canvas. I need to remember to do so. So many things to do. Yeah. So if you want to shoot me an email to remind me, I wouldn't mind. Otherwise, folks, I will speak to you all next week. Have a good weekend. And do remind me to put some of these things on YouTube now, please. I keep forgetting. Especially the stuff here, as I know you're gonna want to revise it, right? So if I haven't done it yet, by next time you see me, just remind me to do it, please. Hi guys.
Sir,
Yes.
one more thing, sorry. As it relates to the YouTube, next class could you send like a link from your channel already? Because when I type in your name on YouTube, I can't find your channel. So
no man, I have to give you a link to the channel.
Yes, I was just asking like next class.
Yeah, man. So what I'll do is I'll post that video, then give you the link to that video, and I think we'll give you the link to the whole channel. And then,
okay, sir.
all right. I haven't actually put anything for this part of the course on there yet. So what I have there is anatomy stuff from the previous one. But yeah, just remind me if I haven't done it, just remind me on Monday about it, and then I'll give you the link, and we can go from there. All right, folks, have a good rest of your night. I want to do the other 3, but whatever. All right, guys, bye-bye.